Above the twilight zone, daylight forms a pale ceiling against the ocean's deepening blue. Anything swimming beneath that ceiling risks becoming a dark cutout visible to hunters below. Yet some animals carry rows of living lights across their downward-facing surfaces. These organs glow toward the depths, filling the shadow that the body would otherwise cast. The strategy is called counterillumination, camouflage built from precisely directed biological light. Its central mystery is control. A mismatched glow could advertise the animal instead. Too dim, and the original silhouette remains suspended against the surface. Too bright, and a luminous patch appears where darkness should have been. The animal must answer changing clouds, depth, posture, and water clarity with adjusted output. If you looked upward through natural twilight, matching brightness would matter more than decorative color. The target is not total invisibility, but reduced contrast across a predator's upward view. Photophores supply the light through tissues specialized for production, filtering, reflection, or direction. Different lineages evolved different arrangements, making counterillumination a repeated solution rather than one invention. Our investigation follows a silhouette through changing light and asks how closely biology can erase it. The opening outline will disappear only where anatomy, behavior, and environment remain aligned. Downwelling sunlight is brightest near the surface and steadily weakens as water absorbs and scatters it. Viewed from below, that residual light creates a brighter background than the darker horizontal water. An opaque fish or squid blocks part of the field and therefore produces negative contrast. Predators with upward-facing eyes can exploit such contrast while searching for prey overhead. Counter-illuminating animals place photophores, mainly on ventral surfaces, where the shadow requires compensation. Many mesopelagic fishes, squids, and crustaceans show patterns consistent with this function. The lights usually emit blue-green wavelengths that travel efficiently through seawater. That spectrum also resembles the dominant color of downwelling light at many oceanic depths. Reflectors and filters can shape emission, while muscles or neural signals may regulate intensity. In some squid, screening pigments move around photophores and alter the light escaping downward. The cue is ambient brightness. The action is changing emission, and the consequence is reduced silhouette contrast. Laboratory and field measurements support this sequence in several well-studied counter-illuminating animals. Functional evidence becomes stronger when output tracks the brightness and direction of natural overhead light. It strengthens further when illumination measurably reduces contrast from a predator-like viewpoint. The Hawaiian bobtail squid, Euprimna scolops, offers a shallow water example with an unusual light source. It houses the bacterium Vibrio fischeri in a specialized light organ beneath its body. At night, bacterial luminescence helps match moonlight and starlight, reaching the seafloor habitat. The squid controls exposure of that light and expels much of the bacterial population each morning. The cue is nocturnal downwelling light. The action is ventral illumination, and the consequence is weakened shadow. This partnership is directly studied, though its setting differs from deep pelagic counter-illumination. In the open ocean, Many species generate their own light chemically within photosites. The repeated geometry remains recognizable: brightness above, darkness below, and a body caught between. Verified context explains the need, but the difficult question is how matching survives constant change. A descending cloud can dim the overhead field within moments, altering the required emission. Waves focus and scatter sunlight, creating fluctuations that a simple fixed lamp cannot reproduce. An animal rolling or turning changes, which photophores face a potential observer. Depth changes both brightness and spectral composition, especially near the lower boundary of sunlight. Some animals respond rapidly, while others may rely on behavioral depth ranges and approximate matching. Photoreceptors can sample ambient light, providing information for feedback control in certain species. The cue is a brighter sky. The action is stronger ventral emission, and the consequence is maintained contrast reduction. Experiments have shown intensity matching, but precision varies, and natural predator vision remains incompletely known. Perfect matching is unnecessary if water scattering blurs edges and predators detect only coarse contrast. Conversely, sensitive eyes at close range may notice individual photophores or gaps between them. Photophore arrangements often break the body's continuous outline into smaller, less recognizable contrasts. This suggests camouflage may combine brightness, matching with disruptive patterning. Researchers measure emitted radiance, compare it with ambient radiance, and model an observer's receptor response. Low-light video can document adjustments, although instrumentation may disturb posture or illumination. Behavioral trials test detection, but captive settings rarely recreate open ocean distances and attack geometry. These limitations separate direct measurements of matching from hypotheses about actual survival. Color matching presents another constraint because a predator may compare spectral as well as total brightness. Blue-green emission is broadly suitable, yet ambient spectra shift with depth and water type. Filters over photophores may narrow or modify emission, improving similarity in particular habitats. No known system makes an animal invisible from all directions simultaneously. Counterillumination mainly addresses observers below, leaving lateral and downward views governed by other defenses. A predator above may see dark dorsal pigmentation against deeper water. Transparency, silvering, schooling, and vertical migration can further reshape the animal's visibility. The cue is an approaching threat. The action may be dimming or extinguishing lights, and the consequence depends on viewing angle. Such shutdown behavior is documented in some luminous animals, but cannot be assumed universally. The evidence, therefore, supports conditional concealment, not a biological cloaking device. That condition connects counterillumination to the daily migrations of immense midwater communities. Many animals remain deep by day and ascend toward food-rich surface layers after sunset. As overhead brightness changes, their required camouflage output changes with it. A fish crossing upward at dusk encounters weakening sunlight, new prey, and new visual predators. The cue is fading light, the action is vertical movement, and the consequence is improved feeding opportunity with managed exposure. Counterillumination can extend the range where that journey remains visually safer from below. For squids, active swimming continuously changes body angle, demanding directional control from ventral organs. Some photophores include lenses, reflectors, or shutters that shape beams more narrowly. That optical architecture spends chemical energy where the concealment problem is greatest. Producing light has metabolic costs, and those costs must be balanced against reduced predation. The benefit likely changes with body size, transparency, habitat depth, and predator community. Bioluminescence also creates social information, allowing signals to coexist with camouflage on one animal. Scientists must therefore avoid assigning every glowing pattern to silhouette erasure. If you see a luminous underside in vehicle lights, position alone does not prove counterillumination. Natural matching requires measurements under ambient conditions and from relevant viewing directions. Artificial white beams can trigger responses, overwhelm faint light, or erase the original background. Careful exploration uses dim instruments and records orientation, depth, and surrounding radiance. Understanding this camouflage improves models of predator-prey, encounters across the planet's largest habitat. It also reveals how evolution exploits photons as structures, replacing missing background light with living chemistry. Future work needs synchronized recordings of animal emission and the overhead field during free swimming. Miniature, low-light cameras could follow vertical movement without flooding subjects with artificial illumination. Visual models should incorporate actual predator sensitivities rather than human brightness judgments. Field experiments could test whether controlled emission changes alter approach distances or attack success. Comparative anatomy may reveal how lenses, filters, and shutters evolved in separate lineages. Measurements across cloudy, moonlit, and moonless conditions would expose the limits of feedback control. Researchers must also determine which photophores serve camouflage, signaling, or multiple functions. Pressure-stable laboratory systems could preserve natural performance in animals brought from depth. The strongest evidence will join radiance matching, behavior, and predator response in one setting. Until then, reduced optical contrast is established more firmly than population-level survival advantage. You can picture the original animal as a dark interruption beneath a luminous ceiling. Then, its ventral lights rise toward the precise brightness surrounding that interruption. Edges soften first, followed by the central shadow, though nearby eyes might still resolve imperfections. The animal remains solid, vulnerable, and visible from directions its lights do not address. Counterillumination erases a silhouette only within a moving corridor of angle, spectrum, distance, and time. That conditional success makes the camouflage more remarkable because it continually negotiates an unstable ocean sky. When you return to the upward view, the missing shadow is evidence of matching, never proof of perfect invisibility.